Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Figure 1.1 is a drawing of a longitudinal section of a human kidney. A is the cortex, B is the pelvis, C is the medulla, and D is the ureter. The loop of Henle is in the medulla, so the answer is C. Bowman's capsule and glomeruli are in the cortex, so it is A for them both. Urine at final concentration will be found in the renal pelvis and ureter. The question wants you to list all the correct letters, so you must write B and D to get this mark. B. Describe the role of aquaporins in osmoregulation. Aquaporins are channel proteins that allow water to pass through. Their presence can increase the permeability of cell surface membranes to water. The target cells are the collecting duct cells. They allow water to be reabsorbed. In other words, water moves into tissue fluid or blood from the filtrate. C. Describe the role of the brain in osmoregulation when the water potential of the blood increases above the set point. To answer this question, you should describe how osmoreceptors work and how they lead to the reduction of ADH secretion. Osmoreceptors detect the water potential of the blood. These receptors are found in the hypothalamus. Osmoreceptors send fewer impulses to the posterior pituitary. This causes less ADH to be synthesized by the neurosecretory cells and released by the posterior pituitary gland. Figure 2.1 shows a recombinant plasmid. It is used to produce IFN-alpha. The plasmid contains the IFN-A2 gene, some regulatory genes, and a gene for antibiotic resistance. A tells us about an investigation to determine how two different inducers may affect the transcription of the IFN-A2 gene. Figure 2.2 shows us the concentration of IFN-alpha produced by the cultures containing the two inducers and a control culture that does not contain any inducer. 1. Explain the role of the gene LEG1 in the control of transcription of the IFN-A2 gene between 0 hours and 4 hours. From 0 to 4 hours, IFN-alpha concentration does not increase. This indicates that transcription did not occur. It shows that LEG1 is a regulatory gene. It codes for a repressor protein that binds to the operator. The binding prevents transcription of IFN-A2 as RNA polymerase cannot bind to the promoter and carry out its function. 2. Describe the changes in the concentration of recombinant IFN-alpha in the culture containing IPTG from when IPTG was added at 4 hours to the end of the experiment at 28 hours. There are two things you should notice in this question. 1. It is a described question, so no explanation is required. 2. You should only describe the IPTG culture. First, provide a general statement. The concentration of IFN-alpha produced peaks or increases and then decreases. Then, describe the peak in the graph. You can get another point by comparing the increase and decrease parts or by comparing the final and starting concentrations. Quote in the data for the point will give you the last mark. 3. Suggest one reason for the difference between the concentration of recombinant IFN-alpha in the culture at 8 hours in the presence of lactose and the concentration of recombinant IFN-alpha in the culture at 8 hours in the presence of IPTG. The command words here is suggest. It means that there are many correct answers. IPTG is higher because lactose has to be converted to allolactose before it can switch on the lag operon. The question also mentioned that, unlike lactose, IPTG is not broken down, so it continually binds to the operon. This also means that IPTG will be at a higher concentration than allolactose. The next point is a logical guess. IPTG may have a higher affinity for repressor protein than allolactose. Since lactose can be used as a respiratory substrate, it can be used up. Its concentration would keep decreasing inside the cell. Lastly, there may be more IPTG entering the cell compared to lactose. 4. Suggest one reason for the change in the concentration of recombinant IFN alpha in the culture containing IPTG from 12 hours to 16 hours. The only reasonable guess is that INF alpha is an unstable molecule. It breaks down in the cell. B. Suggest how AMPR allows genetically engineered E. coli containing the recombinant plasmid to be identified. Antibiotic-resistant genes are frequently used for the screening of transformed host cells. The scientists can expose the cells to ampicillin. The E. coli that have taken up the plasmid will survive since they have the resistant gene. C. Outline how bacteria can evolve to become resistant to antibiotics. You need to use the concept of natural selection to answer this question. A random mutation may have occurred, which led to the arising of the antibiotic-resistant gene. When the bacteria are exposed to antibiotics, natural selection occurs. The type of selection that takes place is the directional selection. Antibiotics act as selection pressure. The bacteria with the allele that codes for antibiotic resistance have a selective advantage. They survive and reproduce. They pass on this allele to their daughter cells by vertical transmission. You can also use the terms asexual reproduction or binary fission. They may also pass on the allele that codes for antibiotic resistance to other bacteria by horizontal transmission. The process is known as transduction, transformation, or conjugation. This is when DNA is transferred from a donor to a recipient bacterium by direct contact. There is a mark for AVP. For example, you can talk about the human behavior which encourages this evolution. Question 3 is about the GM salmon. A. Explain what is meant by continuous variation and how it can be caused. This type of phenotype changes gradually over a range of values. You can also use the word intermediates. You can't categorize the phenotypes into groups, classes, or categories, as they are not discrete and not qualitative. They are quantitative instead. A continuous variation graph shows the normal distribution. There are 2 marks for the second half of the question. It is usually polygenic. Polygenic means it is controlled by many genes. A few different genes that have additive effects contribute to the phenotype together. It is usually affected by environmental factors too. It is not only due to the genotype of the organisms. In B, we have some information about research on recombinant growth hormone. Scientists inject recombinant growth hormone into a group of non-GM salmon. Their mean body mass is compared to a control group without the injection. Table 3.1 shows the result. 1. Calculate the t-value for the results and give your answer in 2 decimals. You must show your work too. X bar 1 and 2 are the mean values for the 2 groups. S is the standard deviation, while n is the sample size. The answer is 7.56. 2 gives you the critical t-value at p equals to 0.05. You are asked to comment on the extent to which the conclusion made by the student can be supported. Notice that this question asks you to comment, not only conclude. So, a conclusion saying that the calculated t-value is greater than the critical value, p is smaller than 0.05, the null hypothesis is rejected, and the difference between the mean values of the 2 groups is significant and not due to chance will only give you 1 mark. You need to comment on other aspects of the comparison to get more marks. For example, the reliability of the test and the validity of the comparison. First, the data range of the 2 groups overlaps, so the difference between them may not be significant, as some of the injected salmon have the same mass as those without the injection. Second, a correlation does not prove causation. We can't be 100% sure that the difference between them is truly due to the growth hormone injection, as we did not use any experiment to prove the connection between them. Besides, the results are obtained from a single investigation. Replicates are needed to increase the reliability of the data. We use the critical probability of 0.05 for the t-test. There is a 5% chance that the difference may be insignificant. Lastly, the t-test may not be valid due to unequal standard deviations. One mean value is more reliable than the other, so the comparison may have a low validity. Question 3 wants you to suggest one advantage of farming GM salmon that produces increased quantity of growth hormones compared to the non-GM salmon that receive hormone injections weekly. It is less labor-intensive, as no weekly injections are needed. This can also reduce the stress experienced by the fish. Weekly injections are a physical stress to them. There are risks of infections with injection if it is not managed carefully. Growth hormones are continuously produced by the fish themselves. GM salmon may grow faster, leading to greater productivity. Question 4a wants you to outline the steps required to prepare the genome of an individual for microarray analysis. Note that this question is not about analyzing the gene expression of a cell, but genome analysis. So, do not talk about extracting mRNA from a cell and making cDNA. In this case, we are supposed to isolate or extract DNA from cells or tissue, rather than mRNA. Cut the DNA into small fragments using restriction enzymes. Then, denature them into single-stranded DNA. Bear in mind that we always use single-stranded fragments in microarray. We must also add fluorescent tags or marker or dye to check the hybridization with the probes later. You don't have to describe the subsequent procedures, as the question only asks you to describe the preparation of the genome. B talks about DGOT syndrome, a dominant inherited disease that is caused by the deletion of a large number of nucleotides from chromosome 22. In figure 4.1, we have the results of the DNA analysis of two individuals, with and without the disease. From region 16 to about 17.3, there is no deletion. Since the DNA from the person with DGOT syndrome fluoresces close to 100% as the control. The same goes for regions 19.7 to 21. Region 17.3 to 19.7 is where the deletion occurs, as the intensity is only 50% of the control DNA. 1. Estimate the number of nucleotides deleted from the affected chromosome 22 in the individual with DGOT syndrome. Since deletion occurs from 17.3 million nucleotides to 19.7 million, the difference between these values is the total number of nucleotides deleted. The answer is 2.4 million. You must give your answer to the nearest 100,000 nucleotides, so write it as the full number, not 2.4 million. Forgetting to round it up may cause you to lose the mark as well. 2. Explain how the microarray technique works to give the result shown in figure 4.1. First, we need to label the DNA from the two samples we want to compare with different color fluorescent tags. Then, allow those single-stranded DNAs to hybridize with probe on the microarray. Sample from the patient with DGOT syndrome would have less DNA binds to probes that control DNA since many of the nucleotides are deleted. So, there is less fluorescence. However, the fluorescence is equal when nucleotides have not been deleted. 3. Suggest why the phenotypes of two individuals with DGOT syndrome can be different. The question mentioned that the number of nucleotides deleted varies between individuals in a range of 800,000 to 3,100,000. So, for different individuals with the syndrome, different numbers of nucleotides may be deleted. The genes deleted may not be the same either. Even if it is the same gene involved, different alleles of the same gene may be deleted. In other words, in one individual, the remaining allele may be recessive, but in another person, it is the dominant allele. So, different proteins are made in different individuals. Lastly, don't forget that our phenotypes are also affected by environmental factors such as diet. Table 5.1 shows some stages in meiosis and their descriptions. You are asked to complete the table. In anaphase 1, Spindle fibers contract to pull the chromosomes toward the opposite poles or to the centrioles. Row 3 shows that two cells have already been formed. The nuclear envelope starts to disintegrate, and spindle fibers are reforming. So, this is prophase 2. In telophase 2, 4 daughter nuclei have been formed. However, the cells are not fully separated since cytokinesis has not occurred. 2 single chromosomes are found in each nucleus as the sister chromatids have separated during anaphase 2. The nuclear envelope is reforming as well. B. Explain the need for reduction division during meiosis. Meiosis or reduction division is important to produce gametes. In sexual reproduction, 2 gametes fuse during fertilization to form a zygote. This resulting zygote will have maternal and paternal chromosomes. So, the gametes must be haploid, consisting of only half the normal number of chromosomes. This will cause the zygote to restore the diploid number. If meiosis is not used to reduce the chromosome number in the gametes, a doubling of chromosome number would occur. This leads to polyploidy, where a cell is having too many chromosomes. Without a reduction division, chromosome number cannot remain constant when fertilization takes place. In question 6, we have a diagram of a mitochondrion. The 4 arrows show the movement of molecules or ions, and you are required to identify them. Protons are pumped actively from the matrix to the intermembrane space, so the answer for 1 is D. Carbon dioxide is the byproduct of the Krebs cycle and the Link reaction. It is produced in the matrix. They are transported out to be excreted, so the answer is C and D. B. Outline the role of the mitochondrial matrix in respiration. The command word is outline, so you don't have to elaborate on each of the roles too much in detail. You should provide a wide range of answers which are short and precise. The matrix of mitochondria is the site of the Link reaction and Krebs cycle. It has DNA and ribosomes to produce proteins used in respiration. Note that if you only mention it is for protein synthesis without linking these proteins to respiration, you will not get this mark. Any named example of the proteins can give you another mark. Reduced FAD and reduced NAD are produced here for oxidative phosphorylation. They are produced in the Link reaction and the Krebs cycle. Lastly, substrate-linked phosphorylation takes place in the matrix to synthesize ATP. It is a part of the Krebs cycle. C. Explain how a lack of oxygen affects oxidative phosphorylation. To get full marks for this question, do not just describe the role of oxygen in oxidative phosphorylation. Your answers should provide the consequences when there is a lack of oxygen. The process would stop or decrease because fewer electrons would be accepted by oxygen. This is because oxygen is the final electron acceptor. ETC will then stop since no or fewer electrons can enter and move along it. Subsequently, no or fewer protons will be pumped into the intermembrane space, causing a less steep proton gradient. You can also say that there will not be a gradient created at all. Without this gradient, no or less chemiosmosis can take place. The stopping of ETC causes no reduced NAD and FAD being oxidized. So, no or less ATP is being produced. There is a mark for AVP. For example, less pyruvate enters the mitochondrion since the process inside has slowed down or stopped altogether. Question 7 shows a synapse between a chemoreceptor cell from a human taste bud and a dendrite in a sensory neuron. The resting potential are -50 and -70 mV respectively. Table 7.1 shows the membrane potential of the two structures when different concentrations of sodium chloride were applied to the microvilli of the chemoreceptor cell. You are asked to explain the results in the table. When 0.1 gram per dm cube was applied, the chemoreceptor membrane was not depolarized. You can tell from the membrane potential, which remains at the resting potential. This means there is no release of neurotransmitters. So, the dendrite of sensory neuron won't get depolarized as well. No action potential is created. For both 1 and 10 gram per dm cube, the chemoreceptor membrane is depolarized. It releases neurotransmitter, so depolarization or action potential occurs in the sensory neuron too. This means that the receptor or generated potential has exceeded the threshold. An increase in sodium concentration does not increase the membrane potential. Positive 30 and positive 40 millivolts are the maximum values for them. This is known as the all or nothing law. B. Describe the differences in structure and function between sensory neurons and motor neurons. Let's compare the structure first. The cell body of a sensory neuron is located between its dendron and axon, while for motor neuron, it is at the end of the cell. A sensory neuron has a dendron and axon, but motor neuron only has a long axon. It does not have dendrons but have smaller fibers called the dendrites. Sensory neurons have no dendrites from the cell body, as they branch from the dendron. Motor neurons have dendrites that extend from the cell body directly. Now the function. Sensory neurons carry impulses or action potentials from the receptor to the intermediate neurons in the central nervous system. Motor neurons carry impulses from the CNS or intermediate neurons to the effector. Note that you should not refer to impulses as messages or signals. A. Describe the functions of the internal membranes of the chloroplast in photosynthesis. You should give a few different functions in your answer rather than just one function and keep on elaborating on this same point. Another thing you should know is that the internal membranes of the chloroplast refers to the thylakoid membranes. They contain photosynthetic pigments to absorb light energy. They are photosystem or antenna complexes and reaction centers. You can also call them light harvesting structures. Photoactivation occurs where electrons are excited, leading to the emission of electrons from the reaction center. The electrons will then move along the electron transport chain. If you name the processes that occur, you can get a mark as well. Those membranes also contain enzymes for the photolysis of water. They are called the oxygen evolving complex. Thylakoids are stacked to form grana to give a large surface area. The space inside is called the thylakoid space or lumen, allows the formation of the proton gradient. It is relatively impermeable to proton and hence can maintain the proton gradient. You can also find ATP synthase on the membrane. Their function is to make ATP by chemiosmosis. B is an investigation on the effect of Rubisco activase on Rubisco. Figure 8.1 shows you the activity of Rubisco in the presence and absence of Rubisco activase. You are asked to describe and explain the results. Let's start with the description. First, give a general statement. For A, the activity increases over time, and B remains low or remains constant. Then, quote in comparative data to support point number 1. For example, in A, the activity increases from 3 to 11.4 arbitrary units, from 0.5 to 6 minutes. For B, it remains at 0.4 throughout. Now, let's suggest some explanations. Tube A shows us that RA activates Rubisco. It may do so by changing the active site of Rubisco. This change may enable Rubisco to bind more readily with RUBP, forming more ES complex. Another possibility is that it causes the product to leave the active site more quickly, and the new substrate can combine to the active site more rapidly. There is a mark for AVP. For example, you can say that RA is a cofactor for Rubisco. It binds to the enzyme and exits its binding to the substrate. Figure 9.1 is a diagram of a relaxed sarcomere. Question 1 wants you to label the actin and myosin filaments. Actin filaments are the thin filaments that are attached to the Z line. Myosin filaments are thick filaments. They join the M line. When the sarcomere contracts, the thin filaments are pulled towards the M line, so the A band will stay the same, but the I band gets narrower. B talks about a toxin that can cause paralysis by binding to the receptors on the cell surface membranes of muscle cells at neuromuscular junctions. Suggests how binding of curare to receptors may cause muscle paralysis. Those receptors are the binding site of the neurotransmitter. The toxin competes with them, so acetylcholine cannot bind to the receptors. In other words, they block the receptors. The sodium ion channel do not open, leading to fewer sodium ions entering the muscle fiber. Without the ions, sarcolemma is not depolarized. This causes no or fewer action potentials to spread across the muscle fiber. Without the action potential, calcium ion voltage-gated channels do not open in the sacroplasmic reticulum, so no or fewer calcium ions are released. If no calcium ions bind to troponin, the exposure of myosin binding sites on actin would be fewer, leading to no or fewer cross bridges being formed. 2. Suggests why the action of curare may lead to the death of a mammal. In the previous question, we have seen that curare can affect the muscle contraction ability of an organism. So, to answer this question, you need to think about the functions of different muscles in a mammal's body and how their failure may cause death. Some examples are the breathing muscles such as intercostal muscle and diaphragm do not work, so the organism cannot breathe. If the cardiac muscle does not contract, blood circulation stops. You can also relate this to heart failure or heart attack. Skeletal muscles that enable movement may be affected, so the organism cannot escape from predators and cannot eat. Question 10 is about biodiversity. Species diversity can be assessed by determining the number of different species and the relative abundance of each species. You can also use the term population. The calculation we will then use is called Simpson's Index of Diversity. Organisms of the same species should have the same genes, but they don't necessarily have the same alleles, which gives variation. Greater genetic diversity leads to a greater ability to adapt or evolve, as not all individuals are affected by the changing environment in the same way. Some might be better at overcoming the selection pressure. Ecological diversity is a measure of the number and range of different ecosystems and habitats or niches within a given area. In B, we have a table showing us the numbers of endangered animal species between 2007 and 2019. 1. Calculate the rate of increase in the number of endangered species. To calculate the rate, you need to find the difference and then divide it over the number of years. The answer is 532 per year. 2. Suggest reasons why more fish species than mammal species are endangered. For this question, please be specific in your answer. All your suggestions must point to these two groups for comparison. Make sure you give a little bit of elaboration when your point sounds general. For example, water pollution is too general. You should provide a little bit more details for this point. Some logical reasons include overfishing by humans for food. Protective laws and regulations related to sea are more difficult to enforce compared to land, so there are fewer laws and regulations in place comparatively. Many people take part in trophy hunting of fish, which means the hunting of animals as sports rather than for food. Climate change may play a part too, but further elaboration is needed. For example, increased ocean temperatures cause predatory species to move into new areas and decrease the number of other species over there. Pollution can be a reason. You can elaborate on this point by referring to the issues of plastic in the sea, oil spills, or eutrophication. There are more species of fish than there are species of mammals, so proportionally, there will be more fish species become endangered. In general, there are more conservation projects for mammals, so they are less susceptible to becoming endangered. Lastly, there are more laws in place banning hunting to protect mammals. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.